Hello everyone and welcome to the scientific overview of synthetic biology. Today we will explore how this rapidly evolving field is redefining our relationship with living systems. We will walk through its historical evolution, major milestones, and its future directions. To understand where we are going, we first need to define what synthetic biology actually is. It is a highly interdisciplinary field that integrates knowledge and technologies from various fields to control and modify living systems. Our ultimate goal here is to build artificial systems that can directly address the many complex challenges facing human society, specifically by driving new possibilities and innovations in healthcare and environmental protection. The foundations of this discipline didn't appear overnight. A great number of significant discoveries emerged in the late 20th century, starting with fundamental genetics. It was during this era that scientists began gradually discovering the regulatory patterns of gene expression, setting the stage for everything to come. Our history truly begins in 1961, when scientists Francois Jacob and Jacques Monod discovered the lac operon in E. coli bacteria. This specific genetic mechanism controls the bacteria's ability to metabolize lactose. A few years after this discovery, restriction enzymes were found in bacterial cells. While bacteria originally used these enzymes to cut viral DNA to defend themselves against bacteriophages, scientists realized their immense utility. These restriction enzymes eventually took on vital roles in constructing recombinant plasmids in later genetic experiments. As we move further into the late 20th century, our ability to manipulate DNA required a way to scale and amplify genetic material. This brings us to our next major milestone in 1983, a breakthrough that completely revolutionized DNA amplification. In 1983, a groundbreaking technique used to amplify DNA sequences was created by Kary-Mullis. This process, known as the polymerase chain reaction, or PCR, allows scientists to make identical copies of DNA strands by mimicking the natural mechanism of DNA replication inside living cells. By using a relatively shorter template strand, scientists can generate a sufficient length of a sequence for future experimental processes. Beyond immediate lab applications, PCR was also fundamental in allowing us to sequence the human genome. Moving on to the 21st century, our understanding of biology was pushed even further by the development of entirely new biotechnologies. This period marked a transition from merely observing and copying genetic material to actively standardizing and synthesizing it. In 2004, the first iGEM conference was held. This launched an incredible platform allowing students from around the world to compete in designing genetically engineered machines using synthetic biology techniques. Just a few years later, in 2008, a massive technical milestone was achieved. The first synthetic bacterial genome was successfully assembled. Consisting of 582,970 base pairs, this marked the largest artificial chemical structure created by the time it was constructed, effectively paving the way for entirely new organisms to be synthesized. By the second decade of the 2011s, the field required more precise tools. This led to a breakthrough in 2012 that transformed how easily and accurately we can edit the genetic code of living organisms. In 2012, the CRISPR-Cas9 system was developed, fundamentally enabling us to modify the genome of an organism with unprecedented ease. Guided by a specific strand of RNA, the Cas9 enzyme acts like molecular scissors, targeting precise sites in the DNA and cleaving a segment off at that exact location. This breakthrough greatly improved all past gene editing methods, making it significantly faster and much more accurate to modify living organisms. This brings us to the present day, as artificial intelligence and computational technologies have begun to flourish in recent years. They have been gradually integrated directly into synthetic biology techniques, leading to an entirely new classification of biological machines. In 2020, an AI program designed the blueprints of a xenobot, a self-replicating and synthetically created biological machine. This milestone beautifully demonstrates the integration of the field, blending cell biology and computer science together to transform abstract biological concepts into programmable functional bioparts. Looking forward, it is highly expected that artificial intelligence will take an even greater part in synthetic biology, guiding complex design and predictive modeling. Under the assistance of synthetic biology, we can transform what used to be impossible into possible, and from possible to achievable. Throughout its brief but dense history, numerous breathtaking advancements have been made by blending cell biology, computer science, and biochemistry. It will undoubtedly continue to be a powerful tool for us to explore the secrets within our genome and the world around us.